Okay, we're going to get started. First of all, welcome everyone to the inaugural memory seminar titled Secrets of the Brain, Neuroscience and Psychology for a Better Life. My name is Blake Beyer. I'm the founder and president of Memory. And I guess I'll tell you guys a little bit about what we do. First of all, we do educational events like these. And second, we raise funds for neurological disease research. Tonight we have two speakers. We have Dr. Thomas Zentall and Dr. Lauren Whitehurst. I want to say I'm thrilled for you guys to be here tonight. I know a lot of you guys are looking forward to pizza dinner. That will be at the end. Next, I want to give a quick thanks to our sponsor for tonight, which is Cats Motivated. Cats Motivated is a club that I'm an executive on. We do events that uplift the community, and we have a big volunteer presence at the hospital. I want to give a thanks to Aiden, and he's also a member of the board of Cats Motivated. Real quick, BB Involved requires us to do attendance, so please scan the QR code to mark your attendance for tonight. All right. Now I would like to introduce our first speaker for tonight, Dr. Thomas Zentall. He's a professor in the Department of Psychology at the University of Kentucky. He is the principal investigator of the Comparative Cognition Lab. He studies comparative psychology, which is the comparison of cognition between animals and humans or across species. He has published over 300 peer-reviewed articles, and his current research focuses on the behavior and cognition in animals, with hopes that his findings can uncover the underpinnings of complex human behaviors such as gambling and addiction. Without further ado, I would like to bring Dr. Thomas Zentall to the stage. His presentation is titled Comparative Cognition, Suboptimal Gambling Behavior in Pigeons.
Is that on now?
Yeah.
Can you hear me? And you have the presenter. Thank you. Okay, I'm not really used to two screens at once, so people over there will have to just follow the pointer. Okay. My interest in psychology is in experimental psychology, and I'm particularly interested in comparative cognition, the relation between cognitive behavior of humans and other animals. The more I study animals, the more I seem to learn about humans, and it turns out that we have a lot in common with other animals, and that tells us something about the origins of our own behavior. For many years, I was interested in the cognitive behavior of animals, and I looked at certain kinds of abilities such as concept learning. So take something simple, relatively simple for us, like same difference learning, when two things are the same or different. If I train a pigeon that when it sees a red light and has a choice between red and green, it should choose the red. If I show them a green light and a choice between red and green, it should choose the green. It can learn that by just stimulus response learning, or it can also learn something about sameness. And I can test that by transferring the pigeon to, let's say, shapes. So I present a circle and then a choice between a circle and a triangle. If the pigeon chooses the circle, it shows that it understands the concept of sameness. Imitation. We humans are terrific imitators. To what extent can animals imitate? And what does that say about how they represent what others are doing? The idea would be, if I do what that other animal is doing, I may obtain the same reinforcer. A third is memory. Cognitive psychologists are very interested in how humans remember. And one of the aspects of memory is how we maintain information in memory to produce at a later time. The question for animals is, do they have the ability to maintain or lose information, maintain information they will need or lose information they will not need? If I tell a pigeon, remember this color, but you can forget this other color, and then later I say, do you remember the one I told you to remember? The animal says, yes. Do you remember the one I told you you didn't have to remember? The animal says, not so well. And what that suggests is that the animal is able to maintain in memory information that it knows it will have to remember at a later time. More recently, I've been interested in suboptimal or biased human behavior. This is where humans behave in a way that's not logical or appropriate, but it shows a bias that social psychologists often think have to do with culture or particular human experiences. One example of this is cognitive dissonance. We've all heard about cognitive dissonance, the dissonance between what you believe and how you behave. Justification of effort is a subset of cognitive dissonance that has to do with the way that we value rewards. And very often we will value a reward more if we've had to work hard to obtain it. The same reward, if it's easy to get, it's not as valuable as if it's hard to get. It turns out that this is something that animals show as well, which is really interesting from an evolutionary perspective. Another one that we've looked at is base rate neglect. That is, low probability events tend to get overestimated. So, for example, many people are fearful of flying because they think it's dangerous, whereas driving to the airport is generally more dangerous than taking an airplane. So we overvalue, we overjudge those very infrequent events that happen, like an airplane crash. And what I wanted to talk to you today about is unskilled gambling. That is, lotteries, slot machines, the kinds of things you encounter at a casino, or lotteries that you buy a lottery ticket. Okay, so this is the question. Do pigeons gamble? And we got interested in this once we started out on a very different question. Just to give you an idea of what this is like, this is a standard operant box for pigeons. Let's see, up here we have 3 buttons or lights. We have a projector here that can project these on these lights, and the pigeon can be trained to peck at them. And if it pecks correctly, down here we get a feeder that provides the pigeon with grain, and then we give them grain because they're hungry. Okay, we began with a somewhat different question that had to do with the value of information. How valuable is information to the pigeon? Would a pigeon prefer information even if the information was not useful in getting more food? Normally we think of information as valuable. So here's the setup. We have 2 lights that come on. We'll call one the information side and the other the non-information side. Oopsie, didn't mean to do that. If the pigeon pecks the information side, there's a 50-50 chance that it will turn on green. And if it turns on green, that means it will get fed 100% of the time. The other 50% of the time it turns on red, and if it turns on red, it means it will never get fed. So green is good, red is bad. On the other side, we have the absence of information. We have blue or yellow, but the blue or yellow does not provide any further information. Either one is associated with reinforcement 50% of the time. That's the non-informative side. And just to clarify, I'm using these colors and these positions so I can keep track of them and you can keep track of them. But when we actually give these to the pigeons, they're counterbalanced. Both location and colors are counterbalanced over the pigeons. So a preference, there would not necessarily be a preference that we find for a particular color. What we found was rather surprising. We found that although the probability of reinforcement is exactly the same, there was a very strong preference for the information side. Even though at the time the animal chooses, it has no idea what it's going to get, there's a 50-50 chance of getting fed on both sides. Okay? This was a bit surprising, but it led us to the next experiment, which was what would happen if we gave the pigeon less food if it preferred the information. Would it still prefer the information even though it gets less food? So here you can see the probability on the left is 20% getting fed. Probability on the right is 50%. Now, what that means is we have a 20% chance of getting the green light, for example, and an 80% chance of getting the red light, whereas on the other side, there's a 50% chance of getting fed just as before. But now we're getting much less food in return for the information. What does the pigeon do with this? What we've plotted here is the percentage of time the pigeon chooses the suboptimal choice. Anything above the dotted line, which is 50%, is a preference for the suboptimal alternative. Okay, so you can see the pigeon very quickly chooses the suboptimal alternative all the time. Keep in mind that they choose, if they choose optimally, if they choose the right side, they would get 2.5 times as much food as choosing the left side, yet they choose the left side. This is beginning to look a little like suboptimal gambling behavior. Okay, now maybe it's not so much that they really like the suboptimal side as they dislike the uncertainty on the right side. What if we gave them a choice where it did not involve any uncertainty? What if we gave the animal a choice in which we manipulated the magnitude of reinforcement? So now you can see that there's 20% chance of getting what we call a jackpot, or 10 pellets of food, versus an 80% chance of getting no pellets at all. And on the other side, what we'll do is we'll give the animal 3 pellets of food no matter what. It has a certain outcome of 3 pellets of food. And if you think about it in terms of, you do the math quickly, you can see that 20% of 10 is an expected amount of 2, whereas on the other side it would get 3, or 50% more if it chose the optimal alternative. And here we can see once again we've got the percentage of suboptimal choice. It starts out, interestingly enough, the animals tend to start out choosing optimally to some extent, and then very quickly choose suboptimally, just as before. Okay, and this is really beginning to look like gambling with pellets. Okay, so trying to make some sense out of this, it appears that the green stimulus in this case, 20% reinforcement, we call that a conditioned reinforcer, has more positive value than it ought to have. Somehow it's very attractive. But the question is, why doesn't the experience with the red stimulus, in which they don't get fed, with conditioned inhibitor, why doesn't that deter the pigeon? Now, it turns out that personally, I don't like to gamble. And I don't like losing. But problem gamblers don't seem to inhibit their gambling when they lose. And this is an example. The pigeon is an example of a problem gambler. The conditioned inhibitor appears to have little negative value. Now think about that. Losing doesn't seem to have the effect that one would think it should have. If the probability of a loss is not important, then a win must be the only important outcome, and the best color wins. So if you think about it, it's not the initial choice that is the probabilities associated with that choice, it's the value of the color that follows. So if we think about it, it's not 20% versus 50%. Instead, it seems to be 100% versus 50%. It's the value of the green versus the value of the blue or the yellow. And similarly, let's see, we'll get back to the pellets. There, sorry, jumped ahead. Similarly with the pellets, it's the 10 pellets versus 3 pellets that seems to drive this effect rather than 2 versus 3. Okay, now we decided to do what in science is called a strong test of a theory. This is a test where what we're trying to do is show evidence of the effectiveness of this theory. Remember, our theory says that it's the color that matters. It's not the original probabilities that matter of the choice. It's just the colors. Okay, but what we're doing here is we're giving the animal a choice between 100% reinforcement, 100% here, yellow comes up, but it always predicts food, and a 50% probability. Now, if you are trying to make predictions about what animals should do, you might say there ought to be a preference for 100% over 50% reinforcement. I actually had a colleague, a biologist, I asked him about that. He said, no, this pigeon is going to choose the 100% side because that's so much better than the 50% side. But our theory says pigeons should be indifferent. They shouldn't care. 50%, 100% doesn't matter. What do you think? Turns out we're both wrong. Once again, proportion of choice is suboptimal. Notice they start out below chance, meaning they're choosing optimally, but pretty quickly they switch. And now at about 70% of the time, they're choosing suboptimally. They're preferring 50% over 100%. So this really lets us think a little differently. So in summarizing some of this, the preference for the suboptimal alternative is determined by the predictive value of the color plus the difference between what is expected at the start of the trial and what is actually obtained. So if you think about it, if you expect 100% reinforcement on the right and you get it, that's good. But if you expect 50% reinforcement, but you actually get 100% reinforcement, that's better. So the probability of reinforcement associated with the choice stimuli is relatively unimportant. Think about that in terms of the lottery. What's the probability of winning the lottery? Who knows, right? Nobody talks about the probability of winning the lottery. Stimuli associated with a loss, no reinforcement, are relatively unimportant. People who buy lottery tickets and they lose doesn't seem to have a big effect on their behavior. The value of the positive outcome is very important. What do they advertise on billboards? I don't understand. $50 million people will maybe buy a lottery ticket. $100 million they probably will, and $500 million for the winner. You get lines of people. I mean, I don't know the difference in money and what you can do with 50 million, 500 million versus 50 million, but it seems to affect people's behavior. The difference between what one expects and what obtains also appears to be really important. So suboptimal choice by pigeons is very much like human unskilled gambling behavior, especially gamblers who have problems with gambling, who are addicted to it. Now, Are there any other ways in which pigeons and problem gamblers are similar? One of the demographics of problem gambling is people who tend to have less will also tend to gamble more. This seems paradoxical, but it's generally true. What about less hungry animals? Will pigeons gamble more or less if they're hungrier? We used a procedure that's a little different. We wanted to slow down the process a bit. 75% versus 50%. It's still, the optimal is on the right side. We took normally hungry pigeons. These are pigeons that get fed once a day. They're maintained at 80% of their free feeding weight, and they're very similar to the pigeons you'll see on campus that are hungry, foraging for food. Then we took less motivated pigeons. We pre-fed them to 92%, which is about as much as we can do without getting, if they feed them anymore, they're not interested, but they're interested and hungry enough to engage in this task. This is normally hungry pigeons. You can see, again, above 50% is suboptimal choice. Normally hungry pigeons, these pigeons started out optimally and very quickly switched. And these are the less hungry pigeons. Okay? They start out right about chance and they choose optimally, at least for quite a while. They're still choosing optimally at the end of the experiment. Okay. What's important about this, by the way, is pigeons that need the food less are actually getting more food. Paradoxical effect. All right, people who gamble a lot report fewer other interests. Now, when you're dealing with humans, it's really hard to look at correlations because you don't know what that indicates. You don't know what the variable is that controls that. But with pigeons, we can manipulate that. So the question is, will pigeons that experience an enriched environment where they have other things they can be doing gamble less? This is our normal housing for pigeons. Pigeons actually are a very good species to work with because they like fairly enclosed spaces. Pigeonholes are actually the way breeders have nests for pigeons to keep them. They're sometimes called rock doves, and they nest in cliffs very often, in little niches in cliffs. And here, 4 days a week, 4 hours a day for 5 days a week, we put them in this group cage with a few other things that might interest them, distract them, whatever. So these are our normally caged birds. They very rapidly choose suboptimally. And here are our enriched caged birds. And you can see there are a couple of things to notice. One is over the first 10 sessions, they're actually choosing optimally. And then they rather get, they are attracted to the suboptimal alternative. And eventually they get to be just like the regularly caged birds. So it's effective. The small amount of enrichment is effective, but not totally. Okay, so overall our conclusions are pigeons too show suboptimal gambling-like behavior. They're attracted to the value of the win. Losing has little negative value, and winning itself has additional value. The idea of winning is when they win, it is greater than the actual value of the win. It's the expectation of not winning and then winning that seems to drive gamblers. The implications of this are that gambling involves basic behavioral processes. When I talk to cognitive psychologists about this, they say, well, pigeons won't behave the way humans do because humans gamble because it's fun. When I talk to behavioral ecologists about this, they say pigeons won't gamble because they forage optimally in nature and they will learn to go for more food. The mechanisms underlying this are the positive value of rewards are overvalued, the negative value of losses are undervalued, and winning appears to have additional value relative to expectations. This is true in humans, and it's true in other animals as well. And these are the people I would like to thank who are involved in this research, and I would be glad to answer any questions you might have. You have time for that?
Thank you, Dr. Zentall. That was a great talk. I learned a lot. I hope you guys did too. Our second speaker for tonight is Dr. Lauren Whitehurst. She's also in the Department of Psychology at the University of Kentucky. She is the Director of Team Science Center and the Center for Clinical and Translational Science, and she is the Principal Investigator of the Cognition, Health, and Sleep Equity Lab. Dr. Whitehurst is a psychologist in cognitive neuroscience who studies the mechanisms of sleep, specifically stress-sleep interactions, and her findings have large implications on the prevention of neurological conditions such as Alzheimer's and dementia, because her research also focuses on the costs of sleep and aging. But I don't want to steal the thunder from Dr. Whitehurst, so without further ado, I'm going to bring her up to the front.
Awesome. Thank you so much. Great. Thank you. Okay, so I need to stand kind of in the middle here. Oh, okay, got it. All right. Hi. Hello. How are we? Good. I am impressed that you are all here on a Friday night. I'll be honest. I am very impressed. Okay, so the title of the talk I'm going to review with you all today is Physiological and Psychosocial Correlates of Good Sleep. And my colleague actually talked to you about like kind of what are the limitations of thinking about correlations. We don't know which one's first and how it's moving and what's causing what. And I'm going to put that up front. That is a limitation of some of the work I'm going to talk to you about today. But that's also some of the work that we have going on in my lab now is trying to use manipulations to change things and intervene to better understand stuff, okay? So we'll talk about some of that. If that's some of your questions, we can get into that in the Q&A as well. Okay, let's get started. So this is what we do know, is that our bodies like sleep. How many of you like to sleep? Right? Yeah, exactly. Our bodies like sleep. And if we don't get sleep, bad things happen to us. So one thing we know is that if you don't get enough sleep, you're kind of more susceptible to heart attacks, myocardial infarctions, right? So that's one thing that we know. In this study, they found that people who were sleeping less than 6 hours had a 20% increase in the risk of having a heart attack, right? We don't want that, right? So sleep is good for us. Also, if you don't sleep, your cognitive function falls to the wayside, right? Like sleep is really good for cognition. We'll talk about some specific ways that sleep supports cognition here in a little bit. But what's really important is that if you don't get enough sleep, in this study, people were getting 4 hours of sleep or less, and they had significantly more risk of having a car accident, right? We don't want that either. And then lastly, this study was actually done by my postdoctoral advisor when I was out in California studying some health psychology. And in this study, what they did, it was really cool. One of my favorite studies. They brought people into the lab, and the week before they brought them in, they looked at their sleep activity. So they had them wear a watch that kind of like a Fitbit, right? To kind of get an idea of kind of how they're sleeping, an Apple Watch, something like that. How many hours they're sleeping on average. Then they brought them into the lab and they exposed them to a respiratory virus. Right? I know, everybody was like, what? Yeah, so they brought them into the lab and then they injected them and exposed them to the rhinovirus. Common cold, not a big deal, right? But they exposed them to it and they looked to see how many people got sick and what their symptoms looked like. How much mucus were they producing? How much were they coughing? Things like that. What they found is that the people the week before that only slept 5 hours or less got more sick. They were more likely to catch the cold, and they had worse symptoms of the cold when they caught it. And so that's what this data is suggesting, is that if you had 7 hours or more, less risk of catching a cold. If you had 5 hours or less, 100% increased risk in catching that cold. So we often think about sleep protecting us from like when we are sick, right? We sleep a little bit more, we're kind of groggy, we need that sleep to recover. It turns out that you need that sleep not to get sick. We're walking out in our world every day, coming in contact with pathogens. If you're well slept, you're less likely to get sick. Okay, this one's a little bit scary. So in this study, this is a study of about 500 women, and they were looking at the likelihood of death, mortality. And in this study, women who were sleeping 5 hours or less across their life died sooner. It took about 7 years off of their life, roughly. Right? That's not great. We don't like that. Okay, but sleep is not just you close your eyes and you just check out for a couple hours. And so a lot of the things that we work on in my lab is trying to just first understand what is happening in our body and in our brain when we're sleeping so that we can think about, well, what's good sleep, right? And how can we get more of it? And so when we think about sleep, sleep's not this monolith, right? We don't just turn off. We're not like a light switch. We don't just power down at night. There's things that happen in our brain and our body that specifically push us to sleep. So there's neurons, particularly in our hypothalamus, that turn on so we can go to sleep. They inhibit certain areas of the brain so that we can get into sleep. And that means that our brain isn't in just one state. It actually kind of cycles through a variety of different states while we're sleeping. So the first thing we know is that we have these cycles of sleep. They last about 90 minutes, sometimes to 120 minutes, but we have about 4 or 5 of them in an 8-hour night of sleep, right, roughly. And so the first thing that we do when we fall asleep is we go into stage 1. Stage 1 sleep makes up a really small percentage of our sleep, maybe 1 to 2 minutes. And it's kind of like a transition sleep. It's like, okay, we're starting to inhibit some of our waking systems. We're starting to slow down some of the neuronal firing that we're having in our brain. And we're transitioning into this deeper sleep stage of stage 2 sleep. And during stage 2 sleep, we have these kind of really specific signatures that we can measure with electroencephalography, which is one of the tools that we use in our lab to measure sleep activity. And so this is kind of what the oscillations in your brain. And what we see is that there's 2 main things that happen that kind of trip our brain from stage 1 into stage 2. The first is a sleep spindle. It's a very fast frequency activity. You can kind of see an example. Do I have a little light? Yeah. This is what that spindle looks like, right? So generally speaking, in stage 2 sleep, your brain is kind of firing at a slower oscillatory rate. So you have this kind of slow activity. And every once in a while, you'll have this really fast burst of activity right here. It's a sleep spindle. It comes from our thalamus in the brain. Anybody know what is the thalamus kind of known for? Anybody? Thalamus? Anybody? Yeah, it sends information through kind of the information from our body, from our sensory organs, gets processed through the thalamus, and then it gets pushed out through up to the cortex. And this sleep spindle is a signature of that potentially happening during sleep. It comes from the thalamus and pushes information up to different areas of our cortex. The other main thing that happens is we have these really big, large waves. We call them K complexes. They are in the delta range, and they are really about a sensory inhibition. So in our lab, if you were sleeping and you're a research participant in our lab, and you just fell asleep, you transitioned to stage 2, and someone slammed the door, and it didn't wake you up, your brain likely shot off a K complex. And that K complex is basically an inhibition signal. It's saying, stay asleep. Whatever's going on out there, don't worry about it. We need you to stay asleep. So again, your brain does certain things to maintain your sleep state. After stage 2, you transition into slow wave sleep, which is what we call deep sleep. If you're in the sleep world, people talk about deep sleep all the time. When I say deep sleep, I mean this. I mean that the neurons in your brain slow down quite a bit. Their firing rate, their action potentials are a lot slower. They're not having as many of them. But when they have them, a lot of places in your cortex have them together. They join them together. So a whole bunch of neurons are firing together and are silenced together. And that gives us this slow frequency, right? But high amplitude. A lot of neurons are firing together and they're silenced together, but they're firing at a much slower rate. So that gives us slow wave sleep. And then rapid eye movement sleep, which I usually think about as like the rock star of our sleep, right? Everyone knows something about REM. It's when we do a lot of our memorable dreaming. But during REM, our activity and the neurons of our brain speed up just a little bit. They fire at higher rates, and the activity as a result in our EEG is a little bit faster. Okay, so not only do we have these different profiles, our sleep also changes across the night. So at first, the first thing that your body really wants to do is get a lot of slow wave sleep. And so we think about that as our homeostatic drive. Our body is driving us back to slow wave sleep, dependent on how long we've been away from it. So it has this kind of homeostatic push, this pressure to fall back into slow wave sleep. So we first get slow wave sleep, these blue blocks here. Once we get enough of that, once our body's like, whew, okay, we're back at our set point, great, we start to have a lot more REM earlier in the morning. So that's why usually if you're waking up around 4 or 5 a.m. from a really kind of crazy dream, that's what that is. It's because you have a lot more REM earlier in the morning. And then in the middle, we have kind of equal parts, a little bit of slow wave sleep, a little bit of REM. And stage 2, as you can see in this lighter blue, is all throughout the night. You have stage 2 all the time. Okay, so not only do we have this kind of oscillatory pattern, we also, in this kind of pattern across the night, we also have certain neurotransmitters that also pattern our sleep. I'm so sorry. I'm doing such a bad job of standing in one place, but I'm gonna try right now. Okay, just stand here. Okay. And so when we're online, when we're walking, we're active, we're awake, we're going throughout the world, you want a lot of neuromodulators, particularly the ones that are going to help you interact with your world. So you have things like noradrenaline, you have things like acetylcholine that helps you with your learning, you have things like glutamate that keeps your brain up and focused. You're bringing a lot of information, so you need a lot of resources. As you fall asleep, those neuromodulators are lessened, right? You don't need to process as much of the information in your world, right? You've done all that. You took all that information in. So you have this kind of reduction during non-REM sleep. But during REM sleep's a little bit weird, right? So you have this increase in firing that's happening in certain cells, and that firing seems to be dependent on acetylcholine. So cholinergic activity is really driving that. So all of these things, all these changes are kind of occurring across your sleep. And because cognition, which is one of the things that I study in my lab, is dependent on some of these different neuromodulatory profiles, we also find that different states of consciousness, wakefulness, non-REM sleep, REM sleep, shape and help support different types of cognitive processes. So when you're awake, right, you have great attention. You can pay attention to stimuli in your environment. You can also encode that information. You can learn something about it, right? When you go to sleep at night and you hit non-REM sleep, that reduction in those neurotransmitters allows you to focus on the information you already have, right? You're not taking in any new information. You're able to process the information that you already have. So non-REM sleep has been associated with memory consolidation. You've learned something through the day. You strengthen that relationship. You make it stronger while you have non-REM sleep at night, so that later when you're asked to do something with that information, it's probed on a test or for some other reason, you can find it. So that's called memory consolidation. You learn something. If you stay awake after you learn something, you will consolidate. If you go to sleep, you'll consolidate better. And also, we have this thing called executive function, this ability to kind of regulate your responses, right? So if you're in the world, somebody probes you to do something, your executive function kind of shapes how you respond and when you respond. Think about like emotion regulation. You have an emotion that occurs. You also have some cognition that might regulate how you express that emotion or how you kind of let other people see that emotion. That's a part of your executive function. And our executive function gets better when we kind of exert executive function. Sleep helps us get better and better at executive functions over time. So you have it, and sleep kind of helps you fine-tune it. And then REM sleep is really good for this creative process. So if you sleep on a problem, right, you've ever heard like sleep on a problem, you'll do it better, right? If you are exposed to something, you can't quite figure it out, I don't really know how to solve this problem, REM sleep seems to be really good for helping you solve a problem that you've already been exposed to. Okay, so here's some data from different studies that have suggested this. So in this particular study, they had a pharmacological manipulation. So they gave people a drug that increased those sleep spindles. What they found is that as they increased sleep spindles, they also increased memory. In this study, that executive function I just described, they found that when somebody learned this kind of working memory task, this ability to kind of hold information in your brain for a period of time and then prompt it later, they did that several times. And what they found was that the amount of sleep in between each of those times predicted how well they were able to do that task. They get better at that task over time. And then lastly, this was one of the first studies that really kind of discussed how sleep impacts cognition. It's an early field, so there's tons of questions still to ask. They found that late sleep that's really rich in that REM activity seems to be really beneficial for implicit processing. So in processing that doesn't require kind of explicit knowledge. Okay. All of that had been known before I got to this field, everything I just discussed. So I showed up and I was like, well, what do I do? And what I was really interested in is brain-body connection. So I was really interested in, the brain is cool. I'm a cognitive neuroscientist, so I had to think the brain's cool. But the brain isn't just up here by itself, right? Like we don't just cut off the neck and have everything going well. we have all of these sensory organs that take in information and provide it to the brain. And the ANS, or the autonomic nervous system, moderates and mediates that process. And so I got here and I was like, well, what's happening in the autonomic nervous system that allows the brain to process this information during sleep so well? And so that's some of the basic data I'm going to talk with you about today. One way to think about the autonomic nervous system is through our heart, right? Our cardiovascular activity is very responsive to the sensory information that we bring in. And so in our lab, we very often measure cardiovascular activity as an indicator of autonomic activity. And so we do that through this method called heart rate variability. I won't run you through the details of it, but it's a fun little method that we use. And prior literature had suggested that you have this shift in autonomic activity across sleep, but they had never connected it to cognition. So in our study, we found, in the studies that we ran in our lab, we found that this shift across sleep happens both during the day, both at night, right? You have this change in ANS activity that drops during non-REM, similar to what you would expect, with a lot of it being made up of parasympathetic nervous system activity. The parasympathetic nervous system is really associated with restoration, rejuvenation. You'd expect a lot of that during sleep. And then you have similar activity between REM and wake. And so we ran some studies that kind of validated that. And we extended it from just nighttime and we moved it into the day. And we said, oh, and you can find the same activity in a nap. Why was that important? It's because if you're only looking at day at nighttime, you might think, is it just because it's nighttime, which would be a circadian effect, or is it because people are actually sleeping? So when you move it into the daytime, you get to say, oh, it's because people are sleeping, because it's happening during the daytime as well. And so that's what we did. So it's sleep, not the time of day, that's responsible for changes in autonomic activity during sleep, which was great for us. And then we ran this paper where we brought people into our lab, about 80 participants. They were quite healthy, young, and we had them sleep in our lab with both electroencephalography, so electrodes all over their scalp, and they had ECG, so we were able to look at cardiac activity. And we had them perform this task. So this task is called the remote associates task. It's typically been used to understand creativity. And how people have used it is they've said that the task goes that you see 3 items, and your job is to find a 4th item that's related to the original 3 items, right? So bass, snow, dance. The correct answer for that would be ball, because you can have a baseball, you can have a snowball, and you can also dance at a ball. So the syntactic relationship between the words is not important. It's just that the word is related to all 3. So the last one is surprise, line, and birthday, and the answer to that is party, okay? So we gave them these items. They're really hard, I'll be honest. They're really, really hard. And then we gave them a really easy task. So we said, okay, after you do that, we want you to do some analogies for us. And these analogies we want you to tell us is the traditional analogy. So like whiteout is to pen, as eraser is to pencil. And we wanted them really to get it right. So we gave them the first letter of the correct answer. So we made it very simple, okay? They took a nap in our lab, because I told you we've already established that the nap is the same. They took a nap, or they stayed awake. Had them take a nap, have some sleep, or they stayed awake. And then in the evening, we woke them up, and we had them do 10 items that they had never seen before, 10 new remote associate items. We had them do 10 items where the answers were exactly the same, so the exact same items that they saw previously, so these are repeated. And then we had them do 10 items where the answers to the item in the evening was the same as the answer to the analogy. And so why is that creativity? Because they had to take this thing that they were exposed to previously and use it in a new way. So that's one of the fundamental characteristics of creativity. You have this thing, you have something a skill that you have. How can you use it to create something new and different? That's kind of fundamentally, so that's what we asked them to do. Previous study had shown that ability, the ability to use this thing in a new and different way, was really dependent on sleep. If you stayed awake, no difference in your ability to do that. If you had a nap that just had non-rapid eye movement sleep in it, so just had that spindle-rich, slow-wave sleep, no difference in your ability to do that. If you had a nap with REM sleep, you could do it really well. And so that's what we did. We didn't have a non-REM only nap. We just had the nap with REM sleep. But we found a similar effect. So we found that for the novel items, it didn't matter if people slept or stayed awake. For the repeated items, it didn't matter if they slept or stayed awake. But for the primed items, when people slept, they performed significantly better, over 40% better when they slept. Okay. So then we said, what contributes to that? What factors make that behavioral pattern? And so we looked at their brain activity, right? We looked at their sleep spindles, we looked at their brain activity. And then we also looked at their cardiac activity, that autonomic activity, say, what matters the most? For our novel items, it didn't matter. That none of those variables are significantly related. For our repeated items, we found that our sleep spindles seem to be associated with this behavior. But when we added in our heart variables, our cardiovascular variables, we found that actually high frequency heart rate variability during REM was the most significant effect. So that parasympathetic activity during rapid eye movement sleep was really related to this performance. And when we looked at it with our primed items, it was even more so related to that performance. So a lot of the behavioral change in that performance was related to that sleep, to that cardiovascular activity during sleep. Importantly, our activity during wake didn't predict performance at all. So it wasn't related to our performance at all. So a clear takeaway here is that this heart rate variability activity, or our measure of the autonomic nervous system during sleep, seems to be more related than traditional EEG features to sleep. So some conclusions for this. Our ANS activity during sleep is a unique predictor of cognitive performance. It's better than some of our traditional sleep features, which is really exciting. And HF-HRV, or this autonomic activity during wake, did not seem to be really important here. Okay. Fortunately for us, we've replicated this a couple of times, looking at different tasks, which has been really great. And then we've talked about kind of some of the mechanisms that might be related to this. So in these kind of several papers, we've looked at some of the mechanisms around this. Why might this be the case? Why might sleep be important for something like that? What we found, there's this really great hypothesis out there around memory reactivation. This idea that when you learn something, your neurons are taking that information and they're strengthening it through their connections, through their kind of repetitive patterns, repetitive communications, repetitive action potentials with each other. They strengthen that relationship. What this hypothesis says is that when a rodent or even a human, that's been done in humans as well, learns something during the day, those neurons reactivate that pattern in which they learn that information over and over again. And that seems to be why or how they consolidate. They kind of hold that information together so that you can have things and have memories throughout your entire life, right? If you only see that thing and can remember it when it's in front of you, that's not great, right? We need something to hold on to it. So this is a proposed mechanism. And what we suggest is that the autonomic activity during that initial learning and then across sleep helps to pattern that reactivation process, helps to pattern how you do that. The ANS is continuously giving feedback to the brain and patterning that response. And so in our lab, we're doing a couple of studies where we're now actually stimulating one of the key nerves that provides some of that input to the brain and trying to manipulate that activity to better understand how it's shaping cognition, sleep and cognition. Okay. A little bit of a transition. How much time do I have, Blake? I'm good? Okay. A little bit of a transition. So that's some of the basic science that we do in our lab. But my lab is called the Cognition Health and Sleep Equity Lab. So we're also really interested in how sleep is patterned across populations of people, not just one individual in our lab sleeping. So this might not surprise you. We are sleepy as a whole population. We are tired. 1 in 3 US adults say that they're sleepy, that they're tired, that they don't sleep enough. And people think this is an underestimate. They think that there's some studies that suggest it's up to 76%. If I'm being honest, that resonates. But it doesn't pattern for the same way for everybody. So if you look, this study was one of the largest studies at the time that looked at this, a group out in Chicago. And what they found is that Black individuals are particularly sleeping less than other racial groups in the US. Significantly less than white individuals. And this is adjusted for a lot of the things, after accounting for a ton of the things that you think might be important, right? After we look at age, look at gender, education. Let's account for their work schedule. Let's account for if they're smoking, if they have hypertension, if they have diabetes, any other health symptoms, right? Let's account for all of that, and this pattern still persists. In a study that we conducted with a couple of collaborators, we actually looked at people all across the US. Each of these little dots represent a different person, and where they lived, the green and purple represent if they're living in an urban area, which is in the green, or a more rural area, which is in the purple. And we patterned this all across we looked at a whole bunch of people all across the US. And what we found is that out of this sample, after looking at a ton of different predictors, sex, education, tobacco use, identifying as Black, having a Black identity was one of our strongest predictors of sleeping less, of sleeping worse, right? Less duration of sleep. So this was like, okay, why is this? What's going on here? So remember, sleep is not a monolith, right? So it's not just any type of sleep that matters. And slow wave sleep, that sleep where all your neurons kind of silence and are excited together, seems to be really important for that memory reactivation that happens during sleep. And so is this slow wave sleep potentially one of the reasons that Black individuals are sleeping less, getting less sleep, and does that matter for their memory? So this study is a study that suggested that slow wave sleep is very important for memory, even across the lifespan. And then this study suggested that Black Americans actually receive less slow wave sleep. So in a study that we conducted in our lab, we asked, does that have a quantitative cost to their memory function? So we brought people into the lab at night. We had them learn a memory task. I'll show you what that task was here in a second. We had them learn the task, test on it. A little bit later, test on it again. They slept overnight with polysomnography, and they tested again in the morning. And we had about half of our participants were Black, and about half of them were identified as white. We had them do this task, a word pair associates task. It's very similar to what you might think about, like kind of fact-based learning for a task, like studying for a test, right? So like flashcards. So we had them look at 2 words, and we told them to associate these 2 words together. 2 words that weren't really related, we wanted them to associate them together and remember them as a pair. And why we gave them those instructions is because the hippocampus, the part of our brain where we encode new learning and kind of do some memory consolidation, it's really good at this. It's really good at pairing things together. So we wanted to really engage the hippocampus. Then they were tested on this again 3 times. So what did we find? We found at test 1, there was no significant difference. Though our Black participants were performing worse on this task, there was no significant difference in their performance compared to our white participants. That test 2, again, no significant difference. Even though Black participants were performing a little bit worse here, it was no significant difference. And again, I'll talk about that difference here in a second. But after sleep, after they went to sleep, that difference became significant. So then, obviously, we have to ask the question, what did their sleep look like? We found that Black participants were having significantly less slow wave sleep, over 20 minutes less slow wave sleep compared to our white participants. This was also seen in proportion of their total sleep. So how much of their total sleep was made up of slow wave sleep? Less. And then those big slow oscillations that I talked about, we looked at those across our racial groups, and what we found was that Black Americans had less of those. So when we ask that question, is that sleep one of the reasons why we're having these racial differences on this memory task? We did find that for Black Americans particularly, sleep seemed to be predicting their memory performance later. For white participants, we saw the same relationship, but it wasn't as significant. So these slow wave sleep disparities in Black Americans might also have these quantitative costs to memory at the daily level. One really important thing is that when we think about why this might matter, is that Black Americans are also at 2 times the risk for Alzheimer's disease and dementia. 2 times the risk to develop this across their lifespan. But often, when we are looking at how to intervene, we wait until individuals are 65 years old to do interventions. These participants were 32, and they're already showing these differences. This is a study where they were correlating these changes in slow wave sleep or slow wave sleep losses with beta amyloid, which is the buildup of toxins that kind of are related to Alzheimer's disease and dementia. And they found that slow wave sleep losses predicts beta amyloid buildup. So this might be one of the reasons why we're seeing this higher rate of this disease in Black Americans. This study also was really important in showing that the trajectory of loss is very different across Black and white populations. What this study found was that after the age of 65, Black and white Americans actually lose cognitive ability, have still the same rate of cognitive losses. Before the age of 65, they show up with they have different baseline performance. So after 65, same trajectory of loss. Before 65, differential performance. So it suggests we have to start looking earlier to better understand these changes that might impact us many, many years later. So I will conclude with a couple of things. Black Americans are at significant risk for poor sleep. Slow wave sleep losses seem to correlate with our daily memory deficits that we see in Black Americans. And waiting to intervene until Black Americans are 65 is probably way too late. Now, what I didn't talk about, what I didn't say, is why Black Americans are getting less sleep, right? So in that, one thing that we do in our lab is we think about where people live, the context, the kind of where you live and what's around you. But we also think about the social context. And social context is extremely important in this particular space, right? Understanding the environment, understanding racism. Sleep is one of those things that's really sensitive to all of those interactions. It's important at the individual level, but it's also very sensitive to stress and environmental conditions. And so why sleep is kind of different and how these conditions are shaping sleep is a future direction in our lab as well. So final takeaways. Sleep is critical for health and well-being. Hopefully you guys will believe that after this. Activity in both our brains and our bodies shape how we learn and remember information. Certain groups of people are more susceptible to sleep losses compared to others, and that might put them at risk for cognitive loss across the lifespan. And early interventions that address race-related health disparities are really desperately needed. These are the people who are amazing in my lab that do all this work that I can't be here and talk without. So every last one of them are important, and I will show their faces as many times as I can. And then thank you all.